Hello students, welcome to a lecture on carbohydrates, classification, chemical reactions and their importance. The main objectives of the present module are to give a comprehensive overview of carbohydrates, to classify carbohydrates on the basis of different properties, to understand the chemical reactions of the carbohydrates, and to help the students to understand different roles and functions of carbohydrates in living organisms. First of all, let's have the comprehensive overview of carbohydrates. Carbohydrates are polyhydroxyaldehydes or ketones or compounds that can be hydrolyzed to them. Carbohydrate is an organic compound comprising only of carbon, hydrogen and oxygen, usually with a hydrogen-oxygen atom ratio of 2:1, as in water. Carbohydrates are technically hydrates of carbon. Sources of carbohydrates are baked goods, commonly contain dietary starch and added sugar. Most dietary carbohydrates come from plants. Sugars and starch are nutritive carbohydrates, meaning they are broken down and utilized by the body, primarily to generate energy. Although dietary fiber is also a carbohydrate, it contributes no calories because it is not digested or absorbed. Grain products are the leading source of carbohydrates in the diet. Grains naturally contain high concentration of starch, which our gastrointestinal system breaks down into sugars. Beans and starchy vegetables such as potatoes, green peas and corn contain high levels of complex carbohydrates that our body digests into sugars. Instead of providing carbohydrates, they also contribute vitamins, minerals and dietary proteins. All fruits and fruit juices contain carbohydrates in the form of natural sugars, such as glucose and fructose. Now, let's talk about some important terms associated with carbohydrates. Stereoisomers. Compounds having same structural formula but differing in spatial configuration are known as stereoisomers. While writing the molecular formula of monosaccharides, the spatial arrangement of hydrogen and hydroxyl groups are important, since they contain asymmetric carbon atoms. Asymmetric carbon means 4 different groups are attached to the same carbon. The reference molecule is glyceraldehyde, which has a single asymmetric carbon. The number of possible stereoisomers depends on the number of asymmetric carbon atoms by the formula 2⁻n, where n is the number of asymmetric carbon atoms. Glyceraldehyde is taken as the reference molecule for the D and L isomerism. The configuration of hydrogen and hydroxyl at the second carbon of glyceraldehyde will form 2 mirror images. They are denoted as D and L varieties. Optical activity. The presence of asymmetrical carbon atom causes optical activity. When a beam of plane polarized light is passed through a solution of carbohydrates, it will rotate the light either to right or to left, depending on the rotation. Molecules are called dextrorotatory or levorotatory. Equimolecular mixture of optical isomers has no net rotation, and hence it is called as a racemic mixture. Epimers. When sugars are different from one another only in configuration with regard to the single carbon atom other than the reference carbon atom, they are called epimers. Example, glucose and mannose are an epimer pair which differ only with respect to C2. Similarly, galactose is the 4th epimer of glucose. Anomers. When D-glucose is crystallized at room temperature and a fresh solution is prepared, its specific rotation of plane polarized light is +112, but after 12 to 18 hours it changes to +52.5. if initial crystallization is taking place at 98 degrees centigrade and then solubilized, the specific rotation is found to be plus 19, which also changed to plus 52.5 within few hours. This change in rotation with time is called mutarotation. Carbohydrates are among the most abundant compounds on earth. They are normally broken down into four major classes: monosaccharides, disaccharides, oligosaccharides, polysaccharides. Monosaccharides. The word monosaccharide is derived from mono meaning one and saccharide meaning sugar. The common monosaccharides are glucose, fructose and galactose. Glucose. Glucose is the main sugar metabolized by the body for energy. The D isomer of glucose predominates in the nature, and it is for this reason that the enzymes in our body have adapted to binding this form only. Since it's an important energy source, the concentration of glucose in the bloodstream usually falls within a narrow range of 70 to 115 milligrams per 100 ml of blood. Galactose. Galactose is nearly identical to glucose in structure except for one hydroxyl group on carbon atom number 4 of the six-sided sugar. Galactose is not normally found in nature in large quantities. However, it combines with glucose to form lactose in milk. The conformation are more stable because their hydroxyl groups are pointed away from the structure, preventing steric hindrance. Fructose. Fructose is a structural isomer of glucose, meaning it has the same chemical formula but a completely different three-dimensional structure. The main difference is that fructose is a ketone in its linear form while glucose is an aldehyde. Upon consumption, fructose is absorbed and converted into glucose by the liver in the same manner as lactose. Sources of fructose include fruit, honey, and high fructose corn syrup. What are the reactions shown by the monosaccharides? Let's discuss them one by one. First is Inderal formation. In mild alkaline solution, carbohydrates containing free sugar groups, aldehyde or keto group, will tautomerize to form indiols, where two hydroxyl groups are attached to the double bonded carbon in mild alkaline conditions. Glucose is converted into fructose and mannose. Second, Benedict's reaction. Benedict's reagent contains sodium carbonate, copper sulfate and sodium citrate. Any sugar with free aldehyde or ketone group will reduce Benedict's reagent and are called as reducing sugars. Third, osazone formation. All reducing sugars will form osazones with excess of phenylhydrazine when kept at boiling temperature. Osazones are insoluble. Each sugar will have characteristic crystal forms of osazone. Fourth, oxidation of sugars. Under mild oxidation conditions, the aldehyde group is oxidized to carboxylic group to produce aldonic acid. Under strong conditions, the first and last carbon atoms are simultaneously oxidized to form dicarboxylic acids known as saccaric acids. Example, glucose to glucosaccharic acid. Fifth, furfural derivatives. Monosaccharides, when treated with concentrated sulfuric acid, undergoes dehydration with removal of 3 molecules of water. Therefore, hexoses give hydroxymethyl furfurals and pentoses give furfurals. These furfural derivatives can condense with phenolic compounds to give colored products. This forms the basis of Mollis test, a general test for carbohydrates. Reduction to form alcohols. When treated with reducing agents such as sodium amalgam, hydrogen can reduce sugars. Aldose yield corresponding alcohols, but ketose forms two alcohols because of appearance of new asymmetric carbon atom. Example, glucose forms sorbitol and fructose forms sorbitol and mannitol. 7. Glycosides. When the hemiacetal group of a monosaccharide is condensed with an alcohol or phenol group, it is called glycoside. 8th, formation of esters. Hydroxyl groups of sugars can be esterified to form acetates, propionates, benzoates, phosphates, etc. Now let's talk about the disaccharides. Disaccharide, meaning two sugars, are commonly found in nature as sucrose, lactose, and maltose. They are formed by a condensation reaction when one molecule of water condenses or is released during the joining of two monosaccharides. The type of bond that's formed between the two sugars is called a glycosidic bond. Let's have a few examples of disaccharides. Lactose. Lactose is a disaccharide formed through the condensation of glucose and galactose. The bond formed between the two monosaccharides is called a beta-glycosidic bond. The alpha-glycosidic bond found in sucrose and maltose differs from the beta-glycosidic bond only in the angle of formation between the two sugars. Sucrose. Sucrose is found in common table sugar and is composed of glucose and fructose linked via a 1,2 alpha-glycosidic bond. Sucrose is an excellent preservative because it has no reducing end or reactive group like the other sugars. Other natural sources of sucrose are found in plants such as sugarcane, sugar beets, and maple syrup. Maltose. Maltose is a disaccharide and it consists of two glucose molecules joined by an alpha-glycosidic bond. Maltose is an interesting compound because of its use in alcohol production. Although maltose is uncommon in nature, it can be formed through the breakdown of starch by the enzymes of the mouth. Now, the next thing which we have to talk about is oligosaccharides and polysaccharides. Carbohydrates that contain more than two simple sugars are called oligosaccharides or polysaccharides, depending upon the length of the structure. Oligosaccharides usually have between 3 and 10 sugar units, while polysaccharides can have more than 3,000 units. These large structures are responsible for the storage of glucose and other sugars in plants and animals. Oligosaccharides. Important oligosaccharides are raffinose and stachylose, composed of repeating units of galactose, glucose, and fructose. These oligosaccharides are of nutritional importance because they are found in beans and legumes because of their unique glycosidic bonds. Raffinose and stachylose cannot be broken down into their simple sugars. Therefore, they cannot be absorbed by the small intestine and are often metabolized by bacteria in the large intestine to form unwanted gaseous byproducts. Polysaccharides. Polysaccharides are complex carbohydrates, are usually monomers and consist of thousands of repeating glucose units. Naturally, they allow for the storage of large quantities of glucose. Starch is the major storage form of carbohydrates in plants and has two different types, like amylopectin. Glycogen is a highly branched polymer of glucose that is the main storage form of carbohydrates in humans. The main chain of the structure is composed of alpha-1,4 glycosidic bonds, while alpha-1,6 glycosidic bonds give rise to the branch points of the polymer. Indigestible forms of polysaccharides are known as dietary fibers and come in many different forms, including cellulose, hemicellulose, pectin, gum, and mucilage. Cellulose and most forms of hemicellulose are insoluble fibers, while pectin, gum, and mucilage are all soluble fibers and readily dissolve or swell when mixed with water. What is the role of sugars in the synthesis of nucleotides? Other sugars of importance are found in nucleotides such as deoxyribonucleic acids and ribonucleic acids. Both RNA and DNA have 5-sided cyclic sugars. However, RNA has one more hydroxyl group than DNA. Glucose 6-phosphate, an intermediate in the breakdown of glucose for energy, can be used for the synthesis of these compounds. Let's try to understand the functions of carbohydrates now. Carbohydrates have 6 major functions within the body: providing energy and regulation of blood glucose. Second, sparing the use of proteins for energy. Third, breakdown of fatty acids and preventing ketosis. Fourth, biological recognition process. Fifth, flavor and sweeteners. Sixth, dietary fibers. Let's talk about them one by one. Carbohydrates provide energy and regulate blood glucose. Glucose is the only sugar used by the body to provide energy for its tissues. Therefore, all digestible polysaccharides, disaccharides, and monosaccharides must eventually be converted into glucose or a metabolite of glucose by various liver enzymes. Carbohydrates spare proteins and prevent ketosis. So why are carbohydrates important if the bodies can use other carbon compounds such as fatty acids and ketones as energy? First of all, maintaining a regular intake of carbohydrates will prevent proteins from being used as an energy source. Gluconeogenesis will slow down and amino acids will be freed for the biosynthesis of enzymes, antibodies, receptors, and other important proteins. Furthermore, an adequate amount of carbohydrates will prevent the degradation of skeletal muscles and other tissues such as the heart, liver, and kidneys. Most importantly, ketosis will be prevented. Although the brain will adapt to use ketosis as a fuel, it preferentially uses carbohydrates and requires a minimum level of glucose circulating in the blood in order to function properly. Before the adaptation process occurs, lower blood glucose levels may cause headaches in some individuals. It's recommended that the average person consumes at least 50 to 100 grams of carbohydrates per day. Carbohydrates as flavor and sweetener agents. A less important function of carbohydrates is to provide sweetness to food. Receptors located at the tip of the tongue bind to tiny bits of carbohydrates and send what humans perceive as sweet signals to the brain. Carbohydrates as dietary fibers. Dietary fibers such as cellulose, hemicellulose, pectin, gum, and mucilage are important carbohydrates for several reasons. Soluble dietary fibers like pectin, gum, and mucilage pass undigested through the small intestine and are degraded into fatty acids and gases by the large intestine. Carbohydrates as biological recognition process. Carbohydrates not only serve nutritional functions, but are also thought to play important roles in cellular recognition process. Dear students, that's all about today's lecture. Hope you have understood and enjoyed well. Thank you and goodbye.